Mitosis and Meiosis in Candyland.
By Jake Knopping and Erin Kersten.
Mitosis alternates with interphase. In interphase, the cell grows and the DNA replicates in preparation for mitosis. The three subphases of interphase are G1, S, and G2 phase. G1 phase is the first growth phase. Chromosomes are decondensed, allowing gene expression. S phase is when chromosomes replicate, each forming two identical sister chromatids. G2 phase is the second growth phase. Centrioles, mitochondria, and other organelles replicate for division and spindle formation begins. Sometimes the cell goes into G0 phase, which is the cell's non-dividing state. Mitosis, or M phase, is defined as eukaryotic cell division that produces exact copies of the parent cell's chromosomes and segregates them into separate nuclei. Here is interphase. After interphase, the cell progresses to mitosis, or M phase. In prophase, chromatin condenses into chromosomes, nuclei and ribosomes disappear, and the mitotic spindle begins to take shape. In prometaphase, chromosomes condense further, and each chromatid develops a kinetochore attached to the spindle fibers. In metaphase, motor proteins move the chromosomes along microtubules to line up on the metaphase plate. The two kinetochores of each sister chromatid face in opposite directions and are joined by microtubules to different centrosomes. Non-kinetochore microtubules interact to push the two chromosomes to opposite poles of the cell. The asters meet the cell membrane, completing the mitotic spindle. Anaphase is the shortest stage of mitosis. Proteins attached to the chromatids are cut apart and sister chromatids rapidly separate. Newly formed daughter chromosomes move toward opposite ends of the cells, pushed and pulled by motor proteins along the kinetochore microtubules. In telophase, two nuclei form and recreate the nucleoli. Fragments of the parent nuclear membrane and other intracellular membranes join to form a new nuclear envelope around each collection of chromosomes. Cytokinesis divides the cytoplasm of daughter cells and chromosomes decondense to complete mitosis, resulting in two identical daughter cells.
Meiosis is a special kind of cellular division that only occurs in eukaryotes. It results in the formation of 4 haploid gametes necessary for sexual reproduction, and it occurs in 2 parts. The first part is meiosis 1. The purpose of meiosis 1 is to separate homologous chromosomes. This starts with prophase 1, where duplicated homologous chromosomes link together in a process known as synapsis. This forms a tetrad or 4-strand structure. Crossing over, or the exchange of segments of chromosomes between homologous chromosomes, may also occur. In metaphase 1, centrosomes move to opposite poles of the cell. The spindle fibers extending from the centrosomes connect to the kinetochores of the chromosomes, which line up parallel to each other along the metaphase plate. Independent assortment also occurs during metaphase 1. Independent assortment is the random way in which chromosomes line up along the metaphase plate. It contributes to genetic variation along with crossing over that occurs during prophase 1. In the next stage, anaphase 1, tetrads split and chromosomes move toward opposite cellular poles, with sister chromatids still intact. In telophase 1, which is the last stage of meiosis 1, spindle fibers and motor protein continue to move chromosomes to opposite poles. Each pole will be haploid at the end of the stage. Then, in cytokinesis, cells split, producing 2 haploid daughter cells with sister chromatids still intact. The purpose of meiosis 2 is to separate sister chromatids. The main difference between meiosis 2 and meiosis 1 is that sister chromatids line up along the metaphase plate and separate during anaphase 2, rather than tetrads separating during anaphase 1. After telophase and cytokinesis, 4 haploid daughter cells are produced, meaning they contain half the number of chromosomes as the original parent cell, which is considered to be diploid.
Mitosis results in 2 identical daughter cells, which are diploid, and is considered to be asexual reproduction. This type of cellular division is common in somatic cells that need to be readily replaced, like skin cells. Meiosis, which is a part of sexual reproduction, on the other hand, results in 4 different haploid daughter cells, or gametes. Another difference is the variation between daughter cells produced in meiosis. The genetic variation in sexual reproduction stems from crossing over, which involves the exchange of chromosomal segments at the chiasmata, independent assortment, and random fertilization.